So control gene expression and operons. So the expression of genes can be regulated in many ways. One of which is by modifying DNA, that should say, excuse me. DNA methylation is a process in which methyl groups are added to DNA to turn off expression in eukaryotes. So methylation can physically block the gene from transcriptional proteins, meaning that actual methyl group that's on it can block RNA and other proteins from actually attaching and being able to read it. And it can also recruit proteins to change its winding patterns around histones. So histones are the little proteins that DNA is wrapped around. And we use methylation to actually affect those winding patterns. If things are more tightly packed, your body's going to have a much harder time reading it. So those are the two mechanisms by which DNA methylation works. Genomic imprinting is another type of gene expression control, and that's when only a single allele is expressed for a particular gene. But it's important to keep in mind that this is an epigenetic process. Epigenetic sort of means above genetic. It can be different from generation to generation, so it's not something that you can pass on. So if you have a gene that is imprinted in you, for example, your offspring may not have that same imprint. It's epigenetic. Their grandkid or their great grandchild may have the same sort of genomic imprinting that you do, but it's not guaranteed because it's not passed genetically. It's epigenetic. So the Lac Operon is almost definitely going to appear on the MCAT. So it's really important that you understand it. It's not super hard conceptually, but sometimes when you look at the picture and you don't understand what you're looking at, it can be kind of scary. So we're just going to summarize all of it. We're going to go over some of the important terms, and then we'll try to apply it to a question or two. So in prokaryotes, regulation of transcription is the primary method of regulation of gene expression. So the Lac Operon is a regulatory mechanism employed by bacteria to control its production of this enzyme called beta-galactosidase, and that's based on the presence of lactose. So we say that the Lac Operon is considered inducible because the product will not be transcribed until the presence of lactose induces its production. So operons can be either inducible or repressible. So inducible means the default state is off, and we have to induce it to turn it on. So it's basically the Lac Operon is just a way that bacteria can efficiently regulate the production of certain enzymes because they need the beta-galactosidase to break down lactose. But if there's no lactose in the environment, then it doesn't really make sense to be producing this because it uses a lot of ATP. So they've developed this sort of negative feedback mechanism, and it's very efficient and it's very energy saving. So the Lac Operon consists of 5 components along with 2 other genes that are located at a distant site. And when I say distant site, I mean like in terms of proximity, actual distance. So the Lac Operon are a bunch of genes that are together, although it is affected by a couple of other genes that are somewhere else, that are located somewhere else in the genome that are not actually close to it. So starting with 1, the P region is the promoter for the transcription of the Z, Y, and A genes, which we're going to go over in a second. Remember, the promoter is just a sequence where enzymes can attach. So if we're talking about transcription in this case, the promoter is responsible for attaching that enzyme. 2, the O region is the operator site where the Lac repressor protein can bind. So operators are really where like activators and repressors bind. So in this case, we have a Lac repressor protein that binds to the O region. 3 is the Z gene, and that codes for the actual enzyme, the beta-galactosidase. 4 is the Y gene, which codes for another enzyme known as permease, and that allows inward lactose transport into the cell. 5 is the A gene, which codes for transacetylase, which is an enzyme that transfers acetyl groups from acetyl-CoA to beta-galactosides, the A gene is actually not, it's not required for beta-galactosidase activity, but it is part of the Lac Operon. So those are the 3 main gene products that come from this operon and that allow the bacteria to make use of lactose if there's lactose present in the environment. The Crp gene is a distant gene that codes for the catabolite activator protein, which we just call CAP, and it helps couple the Lac Operon with glucose levels in the cell. So At the most basic level, the Lac Operon responds to the presence of lactose, but it also responds to the presence or absence of glucose. And the CAP protein, which we're going to talk about in a little bit, does that. The I gene is also a distant gene that codes for the Lac repressor protein, so we can't forget that. It creates the repressor protein, and then based on a certain condition, which we'll talk about in a minute as well, it will bind to the O region or not bind to the O region. So both glucose and lactose levels affect the Lac Operon. In general, an absence of glucose and the presence of lactose will result in transcription of the Lac Operon's gene products. So the presence of glucose in the cell inactivates this enzyme called adenylyl cyclase, which in turn lowers the levels of CAMP in the cell. So whenever we're talking about cell growth, adenylyl cyclase and CAMP always come up. Signal transduction, we've talked about them as well. So it pops up a lot when we're talking about stuff like this. In this case, though, CAP that we were just talking about actually has the complex with CAMP to bind to the promoter of the Lac Operon and activate RNA polymerase. So that's how the RNA polymerase is activated. So high glucose concentration means low CAP-CAMP complexing and low transcription rates, and vice versa. If we have low glucose concentration, we're going to have higher CAP-CAMP complexing, and then we're going to have higher transcription rates. So the absence or presence of lactose will also affect the operon. When lactose is not present in the cell, the repressor protein remains bound to the operator, and transcription cannot occur, regardless of glucose levels. So if you look at this graphic to the right, in any of these situations where there is no lactose, you see this repressor protein is bound to the operon. So it doesn't matter how much or how little glucose is in the environment. If this repressor protein is on here, then there's no way that we're going to be able to transcribe this. So the way it works, lactose acts as an allosteric inhibitor for the repressor protein. So this is sort of like the simple negative feedback mechanism. So when lactose is present, it induces a conformational change in the repressor protein, and that causes it to fall off of the operon, and it allows transcription. So the repressor protein is on, that's our default. Once lactose is present, it's going to find its way to the repressor protein, it's going to bind to it, and it's going to change its shape, and then the repressor is going to fall off, and it's going to allow for more transcription. So the Trp operon is a lot simpler than the Lac operon. And the Lac operon is usually one that we see more often, but definitely you should understand the Trp operon as well. This is pretty much just like a basic feedback mechanism. With the Lac Operon, we were talking about glucose levels and lactose levels, but this is pretty much just dependent on the concentration of one specific substance, and in this case, it's tryptophan. So another bacterial operon of interest is known as the Trp operon, and the Trp operon is a repressible one, whereas the Lac operon was inducible because its default is to stay active until it's repressed. So the Lac operon is off until we turn it on. The Trp operon is on until we turn it off. So bacteria use the genes in the Trp operon to create 5 enzymes that convert charismic acid into the amino acid tryptophan. And just like the Lac operon, we only really need to do this when the bacterial cell doesn't have tryptophan. So it's devised a way to efficiently create tryptophan when it needs it, and that is the Trp operon, which is driven by the presence or absence of tryptophan. So the genes in the operon will be transcribed unless there's a high amount of tryptophan in the cell. So when tryptophan is present, it binds to the Trp repressor protein and then binds to the operon's operator to inhibit the transcription. If there's no tryptophan in the cell, the repressor protein cannot bind, and the transcription of the Trp genes can proceed as normal. So this is a lot simpler. It's just gonna keep on transcribing. It's going to be making those enzymes to form tryptophan. But if we have a high enough amount of tryptophan in the cell, then there's no reason to be doing this. It binds to that repressor protein, and it inhibits transcription because we already have tryptophan in the cell.